interested in genome privacy, but particularly genome privacy of this data group that is sort of overlooked over the years is the functional genomics data. So I hope I don't need to convince you guys that data is important, and more specifically, biomedical data can save lives. So we believe that a foundation of a good healthcare lies on the basic research many of us do in the areas of transcriptomics, epigenetics, and genetics. Because of this feedback and the relationship, now our subjects that we do the research on are becoming more and more real individuals rather than model organisms, for example. And that creates a privacy problem as far as the sharing goes. But we want to share the data because it's important for testing new pipelines, for rapid advancement of biomedical data, biomedical science, and reproducibility, and so forth. But there is sharing creates problems because, especially if this data is coming from DNA of real individuals, then you're giving out private information about these individuals. So traditionally, genome privacy focus on DNA data, the genetic data that comes from DNA, classical DNA sequencing experiments. So over the past decade, we've seen that there's leakage in GWAS studies. You can actually cross-reference people's addresses by looking at their, let's say, personal genome profiles, and you can even infer people's last names from their genome. What I'm going to talk about today is the leakage that comes from transcriptomics and epigenetics data. So transcriptomics and epigenetics data, traditionally, their focus and purpose is to infer biological information about disease versus normal cells, for example. You want to know if a TF transcription factor is bound on a genome that has a disease or not. You want to know what are the important genes for the development of developmental stages. So, but it still comes with the sequencing information. However, you can imagine that it's the sequencing data that comes from these experiments are less abundant in the genome. So if you have a transcription factor that is not bound everywhere in the genome, then the coverage from this data you get is very small. And traditionally, the read lengths are also very small. So for example, if you have a read length of 30 base pair of a experiment from a transcription factor that is not bound to many places in the genome, then you will get very small amount of reads. Then the question becomes, does this data really leak information about the individual? Because with the classical genotype detection algorithms, you may not be able to see these SNVs because there's not enough depth in the data. You have, let's say, 1 million reads with 30 base pair length, so maybe you cannot see anything. So then, but most importantly, that if you look at the life cycle of these experiments, then you may actually think that there might be a lot of leakage, especially when it comes to RNA sequencing data. So if you want to, for example, if you want to know the gene expression in different tissues from the same individual, you take samples from these individuals' different tissues, you send it to the sequencer, and then you overlap all these sequences with known genes, and then you can say in which sample which gene is expressed. And what you want at the end is this matrix of genes versus samples, but it comes with this data, and this might take us back to the individual. So then the question becomes, as I told before, that how much information in these experiments? Because currently the policies regarding to sharing of functional genomics data is very ad hoc. A great example is HeLAS genome. So you cannot find a HeLAS genome. It's locked. You have to have special permission to access the data. But if you want to know about the ChIP-seq of HeLAS genome, you can actually search PubMed and then get the sequencing reads from ChIP sequencing reads from different transcription factor binding or histone modification experiments. Same for the RNA-seq, but it's less of a case for RNA-seq. So if there is information leakage in this data, then how can you protect this information while still sharing this data? So those are the two questions I would like to answer in this talk. So we took an information theory-based approach. the analogy is that if you have a noisy radio channel and you have an input and you get a signal, you want to know what the input is. Then it depends on how abundant your signal is. So if you're hearing a word that is very abundant in your daily speech, like the, then you're not going to learn much about the input. But if you're hearing a word that is not very common, then you might guess what the input is. So using the same analogy, that the functional genomics experiments and the genotyping that you do from these experiments are very noisy because you don't have much coverage. The sequencing is not really as high quality as a DNA sequencing experiments. So you have a biosample, you do functional genomics experiments, and then you get these noisy genotypes. If the genotypes that you get is common in the population, then you don't learn much about this individual. But if you're actually capturing some of the rare variants, then you might learn a lot about this individual. So basically, we quantify the information based on the genotyping frequency of the variant that you genotype from the functional genomics experiment reads. So based on that, we can actually come up with several measures. If you already know these individuals' genotypes, then you have a gold standard. And you can look at the gold standard genotypes and intersect it with the genotypes that you get from the functional genomics experiments, and then quantify the information in that intersection, which is called the pointwise mutual information. And then you can tell, with respect to this gold standard, how much information this experiment is leaking. So what we can do also further, we can actually subsample reads from the chip sequencing or RNA sequencing experiment, and for each subsample reads, we do the genotyping, intersect it with gold standard, and quantify the information. And now we can compare it with the gold standard information that you would get if you have the high quality genotypes. So from this plot, what you can say, it's not surprising that as you sample more reads, you will get more information. It's also not surprising that high-depth experiments like HiC will give you better genotypes compared to experiments like single-cell RNA-seq or ChIP-seq. But this still doesn't tell us that if this information is enough to link this individual to their full genotypes, or what can you learn from these little amount of information you get, for example, from single-cell RNA-seq data. So what we did is we designed a very simple linking attack scenario where you have these low-depth experiments and you also subsample the reads, so you have very low coverage. And then you do the genotyping, and you have a, let's say, a genotyping panel with a lot of individuals in it. So what you do is you use every single individual's genotype in this panel as the gold standard, and then you intersect with the genotypes you got from functional genomics experiments, you quantify the pointwise mutual information, and then the highest ranked pointwise mutual information belongs to the individual that you are querying. So, and especially if this individual's PMI value is a lot different than the rest of the population in that genotyping panel. So what we are showing here that even though these low coverage experiments like single-cell RNA-seq or ChIP-seq, even though they don't leak much information, you cannot use it for high-quality genotyping. You can actually link these individuals to the databases by using this small amount of information you get from them. So then, furthermore, what if this individual is not in a database? What can you learn by looking at these chip sequencing or RNA sequencing experiments? So what you can do is you can take all the publicly available ChIP-seq and RNA-seq data, you can put all these BAM files together, do genotyping, and get some number of genotypes. And then on top of that, you can use the population LD structure and impute the rest of the genotypes, and you can actually assemble almost full set of genotypes of this individual by using these experiments. And you can even infer some private phenotypes by just looking at the reads from these experiments. So I guess there is no question that these experiments, the reads from these experiments leak private information, even at very low coverages. But we believe that the sharing of such data is very important for reproducibility, for the advancement of biomedical data science. So then we came up with some means to protect individuals' privacy while being able to share this data publicly. And we created this file format manipulation called price-aware binary alignment map based on the known BAM and SAM file formats. So basically what we do is we use suppression and generalization for certain columns of the BAM files that you don't need to have those columns for the quantification of gene expression, for example. They are just there because you get it from the aligner or from the mapper. So you can actually suppress or generalize these columns, and you can still quantify transcription factor binding or gene expression. But when you do that, because you're suppressing and you're suppressing some of the fields, you are actually adding some noise to the data. So to quantify the noise, what we do is we look at the gene quantifications or TF binding quantifications from the real BAM files, and that versus the gene quantifications from the PBAM files. And then this difference is quantified as something called epsilon, and if your epsilon is very close to 0, then you know that the utility of these files are high. So we actually quantified base pair resolution and exon resolution signal profiles from BAM files and the PBAM files by removing certain type of variants. For example, we can remove variants that are that have certain minor allele frequencies, or we can remove all the split reads, or we can remove just mismatches. You can choose what to remove, or you can remove everything. And then, as you can see in this plot, that our epsilon values are very close to 0, so that these files have high utilities while protecting the sequence information from the people that you're distributing to. So this new file format or file manipulation system works well with many functional genomics pipelines, including STAR for signal tracks, RSAM for gene expression quantification, and MAX2 for ChIP-seq peak calling. And also, we created these software suite called P-tools, where you can take a BAM file, create a PBAM file, and create these small files called .diff files, where you put all the sensitive information, but they are a lot smaller than the BAM files. And then you can go back and forth between BAM and PBAM files with, if you have the reference genome that you mapped this data to. So basically, you don't need to store the original BAM file, which is large in some cases. You can just store this small .diff file, lock it up, and then use the PBAMs for your quantifications. So with this, and with the previous work that we did in the lab, we can now quantify private information leakage at every step of the functional genomics data processing. So we start, in this talk, I talk about the leakage from the reads itself. We showed this here that there's leakage in the signal tracks, and then we showed previously that there's actual leakage at the gene quantification levels. So you can see that we can tell how many number of potential SNPs you can leak from each set of data. And we created this triangle where here in the bottom of the triangle, the utility of the data is high, where you have high private information leakage. As you go up, the utility goes lower because you have high level information about the experiment, but then there's not much private information leakage. So our modified read sets in a good place where you get high utility and also basically utility and privacy balance. So with this, I'd like to thank the members of your STEM lab, specifically Mark, Arif, Molly, and Fabio, and our collaborator, Professor Brenner from UC Berkeley, BD2K and ISCB for bringing me here, and BD2K for the support. And also, our lab is hiring postdocs. If you're interested in genome privacy, please apply. And also, if you have work in genome privacy or biological data privacy that you want to share with us, please consider submitting to PSP. We are having a session on genome privacy. Thank you.
Other questions? Please come to the microphone. Okay, I have a question. So the PBAM, so often when we do gene quantification, we would like to filter maybe by a number of mismatches and so on. So I guess you lose that ability to do that, right?
You lose that, so you have to do it when you're converting from BAM to PBAM. Okay. So for that you need to access to the BAM files, yeah.
But if I would provide multiple versions of the PBAM file with different filters, would that give me again the leakage? Would that leak the information again?
Yeah. So for example, what I did here, I removed, for example, in the last case, I removed all the split reads, right? So it's basically a certain number of mismatches. They are all removed. But then you end up with a lot of mismatches and small indels. Then that will still leak information. So I think it's not safe unless you remove everything, all the mismatches, indels, and split reads.
But I guess what I'm wondering is, if I look at the epsilons, I could probably reconstruct where the mismatches are, and that again is what you don't want.
That's possible, yes.
Yes.
That is possible. But you need to have the BAM file to look at the epsilons, right?
But if I have multiple PBAM files, I can quantify each of them.
Oh, I see. Different degrees. Yes, you can do that. Yes.
Right. There's a question.
Yes. Yesterday I heard some talks on the MPEG-G format that's coming forward. I'm just curious if there's any correlation between this. Will that suffer from some of the same leakage, or how that?
I don't know that format, but the reason why we did PBAMs is because we wanted the file format to base on BAM and SEM files, so that because there are already a lot of pipelines out there for functional genomics experiments, so we want people to be able to use their existing pipelines with a file format. So if you create a different file format, then you have to change the code. So that's why we based it on BAM and SEM files. And you can use it with SEM tools as well, so that's also another advantage.
Thank you.
Good. Thanks so much.